Hello friends, welcome back to another video tutorial from Shomu's Biology. And in this video, I'm going to talk about one of the most burning topic of discussion in modern molecular biology, that is CRISPR-Cas9 system. We're going to talk about all about CRISPR-Cas9 system, how CRISPR-Cas9 system works, what are the important regions of science where we can use CRISPR-Cas9 system. So watch this video to understand, and let me open the Pandora's box that is CRISPR-Cas9 genome editing system. So the very first thing that I want to talk about regarding the CRISPR-Cas9 system is that I already made a video earlier regarding the CRISPR-Cas9, but today I'm going to talk a little detail about the CRISPR-Cas9 system. Now, before starting to talk about the CRISPR and Cas9, first things first, what is it? Because this is a big fuzz, and there are Nobel laureates regarding CRISPR-Cas9 for the last few Nobel Prize. If you see last few Nobel Prize in physiology, they are going to get it from CRISPR-Cas9 because it's a modern pace of research. So what is CRISPR-Cas9 in reality? The thing is, CRISPR-Cas9 system is, in very simple term, a genome editing system. So I can simply write it as a genome editing system. Okay, genome editing system means it's a kind of system which can destroy the targeted DNA which is present inside a cell. Now that cell can be eukaryotic cell mostly, and in eukaryotes that can be lower eukaryotic organism like flies, fruit flies, and also higher eukaryotic organism like us. That's why it makes more sense. So the CRISPR-Cas9 is a genome editing system. That means this CRISPR and Cas9 can cut a target DNA in specific location. So I have a specific activity of cutting a DNA in specific location, and after cutting that DNA, we can render that DNA totally destroyed, or as a result of the cut, our own body's defense mechanism, also the DNA repair mechanism, try to fix that cut, and during the fixation of that cut, it is error prone process, and as a result of that, it may introduce some sort of mutation, some sort of altered nucleotide into the genome, and due to this mutation and change in the organism, we can modify our target gene in many different ways. Okay, so in very basic terms, what I can tell, if I can divide this CRISPR-Cas9 these two terms, what we get is this. We get first thing, that is CRISPR, and the second one is Cas9. These are the two components of this system, right? Now, the very first thing, let me tell you about the idea about this CRISPR and Cas9. The thing is, this CRISPR is basically an RNA structure, a small segment of RNA structure, where Cas9 is a protein. What kind of protein? A nuclease. A nuclease protein can cleave a nucleotide sequences. It's a deoxynuclease, so it can cleave the DNA segment or DNA sequence. So it carries an RNA template, okay, and this RNA template, and the Cas9 contains a protein which is a nuclease. Now the idea is very simple. I can give you a small demonstration of what this CRISPR-Cas9 can be capable of doing. The thing is, normally what we found out is that in this particular video, I'll be explaining in the step-by-step manner about the different things. So I'll first talk about the history, okay, and the origin of CRISPR-Cas9. Then I'll talk about the mechanism of how it works, and the third thing we'll talk about is the use of the CRISPR-Cas9 in future times. So let's first talk about the origin of CRISPR-Cas9 system. As I told you, that this CRISPR-Cas9 system is a very basic, very simple idea, but has enormous capabilities. The thing is, it has an RNA template and it also has a nuclease. Now the idea is how this CRISPR-Cas9 works. Let's assume that we have a dog and we have a bone. What we can do, we can throw the bone and we want the dog to pick the bone. So dog will go and pick the bone because the dog loves the bone, right? So you have a bone, you throw it up, your dog picks it up and brings it to yourself. So this is the idea about CRISPR-Cas9, pretty much. That in this case, this CRISPR is the bone and the Cas9 is the dog. So thing is, the Cas9 can cleave nucleotide sequence. Cas9 is a nuclease, so it can degrade or destroy DNA, double-stranded DNA sequence. But a dog will only pick the bone if you throw the bone. So in this case, the bone is the CRISPR. Okay, so CRISPR is the template DNA, the target, the template DNA which is provided, and that template DNA need to match the complementary sequence with the target DNA which we need to cleave, right? So what we have a target DNA. Let me draw here. We have a target DNA. This is the target DNA, okay? And there are specific nucleotide sequence which is known as the target nucleotide sequence, which is complementary to this CRISPR RNA that is present in there, okay? So what happens is actually simply this CRISPR RNA can interact with this target DNA, okay? It can bind with the target DNA because of the complementary structure that is present between the this CRISPR RNA as well as the target DNA strand. So upon this binding, this cas9 is activated, and cas9 is always associated with this CRISPR. So this cas9 has the catalytic domain, and that catalytic domain simply can cut this target DNA strand, both the DNA strands. I must draw both the strands in here like this. So it will cut both the DNA strand at a particular location, and as a result of which what we get a target DNA completely broken down from the center point, completely destroyed from the center point as a result. So this is the idea. When we break the DNA, that means any information which was present here in the DNA is lost. Now what happened is that after the DNA damage, our DNA repair mechanism in our body try to fix it. And this is a double-stranded break, remember. And if anywhere we have a double-stranded break, then we need to use two methods of DNA repair. One is the double-stranded break repair model, that is a non-homologous end joining or a homologous recombination. Now both of this process, the homologous recombination and non-homologous end joining, particularly the non-homologous end joining, this is error prone. That means the process may conduct, but it may add some erroneous nucleotide in this position, because some of the nucleotides may be lost, as a result of which at the end what we get is a DNA which is probably intact, but that DNA may contain some of the nucleotides in the center which is not wanted. So something totally different. So this lead to different kind of modification in the host organism. What kind of modification? Let's say due to this nucleotide change, the pattern of RNA transcription is altered, and due to this nucleotide change, as the pattern of RNA transcription altered, the protein synthesis is also altered. It can also happen that they can add, due to this addition of erroneous nucleotide, the RNA that they are producing is truncated version. So the protein that they will produce will be a truncated version of the protein, and the protein will be malfunctioning. On the other hand, it may also lead to the origin of a stop codon. As a result, a small copy of the protein is produced with respect to the large amount of protein, the big protein that is supposed to be produced. Okay, so this is what is happening here due to this CRISPR-Cas9 system. So this is pretty much the basic idea. So here we have the Cas9 as a dog and CRISPR as the bone. So what we need to do, once this thing is clearly understood, we can design this CRISPR RNA in such a way, let's say whatever target we have, whatever DNA we can use as a target, we can make a complementary RNA structure of that, and we can tag it with Cas9, and then CRISPR and Cas9 complex will be loaded automatically. They will scan for that complementary structure of CRISPR to the target DNA, and whenever they find it, they will stop there and they will cleave the DNA in that position, and that opens up the Pandora's box, right? Because we can targetedly destroy the DNA, break the DNA, and not only break the DNA, but also we can add some erroneous nucleotide to the DNA, so the total DNA content can be changed. The function of the gene can be analyzed with this. Or if we tag this CRISPR-Cas9 system with some sort of fluorescent tag, we can also mark specific region, specific DNA in the whole genome of a complicated organism like us. Okay, that's the basic idea. But now the question is how and when this CRISPR-Cas9 idea was developed. It was developed very early, but the thing is, modern, like from the 1990s or 92, that from that time onward, the idea about the CRISPR-Cas9 even get much better. The thing is how the CRISPR-Cas9 originated. The idea is this CRISPR-Cas9 system depends on the idea of the bacterial immunity. Okay, we always talk about immunity of human, right? We always talk about our immunity, but bacteria, which is also pathogen, has its own immunity. Okay, because bacteria can be attacked by phage virus, right? Bacteriophage virus. So bacteriophage virus itself is a pathogen of bacteria, pathogen for bacteria. So to protect themselves from the phage attack, bacteria utilizes this cas9 CRISPR mechanism. Okay, and that CRISPR Cas9 mechanism, once we found inside of a bacteria, we are trying to replicate the same process in vitro studies, and that's what we are doing. The thing is, normally what we found out is that this is a bacterial cell, okay, and in this bacterial cell, a phage is attacking the bacteria. Let me draw the phage structure here, like this, okay. The phage is attacking the bacteria, and the phage is transferring its its genetic content inside the bacteria. And let's say this is bacterial chromosome inside, okay, circular bacterial chromosome. So this is phage, this is bacterial chromosome. Now what will happen is that in this phage DNS, there are some places where we have this sequences, okay, these sequences of what? This CRISPR kind of sequence. Now the thing is, the bacteria, now normally this phage can destroy the bacteria, it can kill the bacteria during the growth of the bacteriophage. What bacteria is doing here is simply, bacteria, we all know that bacterial chromosome is always ready to be in contact with some plasmid or any other extra chromosomal structures from outside. It can fuse with it. The same thing happens here. The component of the phage DNA can be fused with the bacterial chromosome, and whenever it is fused with the bacterial chromosome, those regions, those points of this CRISPR DNA system, the CRISPR structures, is already present inside the bacteria. Now what bacteria is doing with the help of this is that this region is the region of phage. Now let's say this bacteria is somehow killed, maybe killed, may not be killed. If the bacteria survives, in the future times, if the same virus comes in and attacks this bacteria, then the bacteria already have its immunity. It's kind of an adaptive immunity for the bacteria, just like we have the adaptive immunity. For example, a bacteria attacks us, so we have antibody build up against that bacteria. From the next time onward, if the same bacteria attacks us, then the antibodies are already ready and they will be fighting against the bacteria. The same thing this bacteria is doing in case of their pathogenesis conducted by the phage virus. So in the next time, whenever the phage releases its genetic material, the bacteria already has their genetic material as a copy, as a memory inside of them. So what they can do is that now they utilize that part of the DNA, and what they can produce is the RNA copy. Okay, it can produce an RNA copy, and this RNA copy itself will be termed as this CRISPR RNA or cRNA. Okay, this CRISPR RNA that is produced, this CRISPR RNA is produced along with a cas9 protein, which is also produced by the bacteria. So this cas9 and CRISPR both together form a complex, and once they form this complex, then that complex is going to interact with this specific region. Try to understand this concept very clearly. We know that the viral region that is coding for the CRISPR RNA is attached with the bacterial chromosome. So if that side of the bacterial DNA, that template of the bacterial DNA is utilized to make an RNA copy or the CRISPR RNA structure, that CRISPR RNA structure can go and can bind with this viral complementary structure, okay, because this RNA has a complementary structure to the viral CRISPR system from where they gain this template in the very first place. So the RNA can interact with that and can bind with it perfectly, sometimes not perfectly 100%, but they still bind with it, and this interaction triggers the activity of Cas9, the nuclease activity of Cas9, and Cas9 will cleave the viral DNA. So as a result of which, what will happen? The viral DNA is cleaved, so the virus failed to replicate and the virus failed to kill the bacteria. So this is a bacterial own adaptive immunity or adaptive immune process. And now what we are doing, we are mimicking this process. We are taking this process and we are taking the advantage from this process. Because when we found out is that the bacteria is killing the virus, the thing that scientists always thought about, how can bacteria kill the virus in the second time? The reason behind it is very clear later, that the bacteria already has that same complementary feature or complementary strand embedded in their genome. So they use the complementary RNA to degrade the bacteria, to degrade the viral DNA. So this idea is utilized further in the mechanism that we are going to see as a CRISPR-Cas9 mechanism, And now what we do, we know that in this system, Cas9 will do the job of cleavage and breaking down the DNA structure. So what we need to do, what we need to modify is this CRISPR RNA or cRNA. So if you modify the cRNA, or if we can provide the CRISPR RNA from outside, if we can provide a complementary RNA from outside. For example, let's say this is our target DNA, which has something like this. So if you want to destroy this particular region, if you want to have a destruction for this, what we can design as an RNA, we can produce an RNA that contains the complementary structure, that will be U, C, C, U, G, U. So this should be our CRISPR RNA. So we provide the CRISPR RNA along with the Cas9, and we allow them to go and bind with the complementary target. This is the target DNA, and this is the CRISPR RNA. So it will bind with it, and RNA DNA hybrid will form, but there the Cas9 protein will destroy this particular target DNA. Not only destruction of a single strand, but the Cas9 can destroy both the strands, degrade both the strands of the DNA. This is the idea. Okay, so now let's talk about the mechanism of how exactly CRISPR-Cas9 works. Now the mechanism. Now this is what I draw regarding the mechanism. Okay, so what we can clearly see is that this is the DNA, this is the target DNA, okay, that we are looking at here. This is the target DNA that we are looking at, okay, and this is the region where we want to cleave this target DNA. Okay, so cleave a target DNA, we need to have the structures. Okay, so particularly what we need to have a CRISPR RNA. The CRISPR RNA is denoted with cRNA. This is how CRISPR RNA is written. Okay, so CRISPR RNA is sometimes used alone, along with Cas9. Okay, along with Cas9, or sometimes this cRNA or CRISPR RNA is also working with TRCR RNA, or we can say as a tracer RNA, trans-activating CRISPR RNA. Okay, along with Cas9. These are the two different situations. Actually, when we talk about this CRISPR-Cas9 system, this is a modern field of research, so new information is coming in every single day. That's why I told you to take the course. The idea is that first there are different types of CRISPR-Cas9 pathway that are being recognized. One of this type 1 pathway that only involves CRISPR RNA and Cas9 nuclease, and there is type 2 CRISPR RNA system where we have cRNA or CRISPR RNA plus Cas9, but one thing extra, that is the tracer RNA. Okay, trans-activating CRISPR RNA. Now, how this process actually works? In case of this type 2, this is the modern one that we want to discuss, the new mechanism that we now know. So this is the target region of the DNA. This is the target DNA. So we know that the Cas9 will be there. So let me draw Cas9 protein. Let's say this is a big Cas9 protein. Okay, and they have a specific catalytic site, and let's say this is one catalytic site, this is another catalytic site. And the catalytic sites are special because they have a sequence known as NGG. Okay, N means any nucleotide, and then 2 G sequences are there. That is the target region of cleavage. Okay, so particularly as I told you that this Cas9 is a nuclease, it's a scissor, it's a molecular scissor, it can cleave. So it cleaves to this specific region NGG, this region, okay, which is also known as PAM site. This is known as PAM site. This full form of PAM site is that protospacer adjacent motifs. Protospacer adjacent motifs. So this is the location which has a particular structure, that is NGG. This particular structure they have. This NGG is a specific sequence where the NGG, N means any nucleotide, and 2 G nucleotide will be present side by side. This is the place where this Cas9 will cleave, because Cas9 is nothing but a molecular scissor that can cleave both the strands of the target DNA. So here what we can see, now you have a question maybe that what is protospacer then? If it is a protospacer adjacent motif, then what is protospacer? Protospacer is the stretch of the DNA where the Cas9 and CRISPR RNA binds. Okay, so this is the protospacer here. Protospacer. This is the protospacer region. The region where the process, the stage of the DNA where the cRNA will bind is known as protospacer. Okay, and that cRNA, which is near about 20 nucleotide long, has complementary structure to the protospacer is known as only spacer. So this is known as spacer element. And so the red color structure you can see, this is the Crispr RNA 5' to 3' direction Crispr RNA 20 nucleotide long. This is known as a spacer element, and the protospacer is the complementary DNA structure to the cRNA, that is protospacer. So now these all structures, you know, we have a protospacer, we have a spacer, we have a cas9, this big cas9 protein which has the PAM site, okay, protospacer adjacent motif where they will cleave the strands of the DNA. So as I told you that this is the type 2, there are different versions and types of CRISPR-Cas9 mechanism. This is type 2 Cas9 CRISPR mechanism. In type 2 mechanism, we not only have CRISPR, RNA and Cas9, but along with that we also have this other stretch of RNA. This stretch of RNA here is known as we already write down here, the tracer RNA. Okay, the tracer RNA is trans-activating CRISPR RNA. This trans-activating CRISPR RNA has this 5' to 3' structure like that, and for the type 2 CRISPR-Cas9 system, both of them are linked by the interlinking loop. So this is a loop that is connecting the cRNA and tracer RNA together. Okay, this is the structure. So normally this is the overall structure of the CRISPR and Cas9, and once they find the complementary region, then what will happen? RNase 3 will cleave this loop out. Remember that RNase 3 has a role, it will cleave, RNase 3 will cleave this loop out, as a result of which what will form? We have this kind of structure where the tracer element, actually the idea of tracer element is not clear, how it is loaded and what is its role is not yet clear, but it was found out that tracer element has some sort of role in stabilizing the CRISPR RNA with the complementary pairing with the target DNA strand. That's why the tracer RNA is required. So once the requirement is done and proper positioning of cRNA is done, then the loop is cleaved by the RNase 3. After the loop is cleaved, then this particular structure, this Cas9, remember this is Cas9 protein, a molecular scissor. Its PAM sites are activated where the catalytic units are there. So one catalytic unit here, another catalytic unit here, which will cleave the DNA from this specific location. So DNA cleavage from this specific location will be done after the job of RNase 3. And that thing will be done by by the Cas9 in the PAM sites. So as a result of which what we'll have, we will have the DNA cleaved, double-stranded cleaved structure of the DNA. Okay, this is the mechanism of how CRISPR-Cas9 work together, sometimes also along with the tracer RNA as well. So once this cleavage is done, now comes down to the very important point where we are going to talk about the use. Okay, so now we are going to talk about the use of CRISPR-Cas9. Remember, after the breakage, so in very simple terms, if I tell you like what is the importance of CRISPR-Cas9, beyond this point what you can say, that yes, it can destroy the target DNA. So if you want to destroy a DNA, if you want to knock out any gene or knock down any of this gene, or we want to silence any of this gene, we can use this specific cRNA that we design in vitro and we provide it inside the cell, and they can cleave the target DNA. That is fine. That is about the gene silencing. This is one of the important jobs, is the gene silencing. Okay, this is one of the important job of CRISPR-Cas9 system, probably the important job that was discovered the first. Now we discovered many more. The thing is that as there is a double-stranded DNA breakage, so that can be fixed with either homologous recombination or non-homologous end joining, where both these processes are complicated and error prone. So they may lead to some sort of error there, some sort of nucleotide in between which is not supposed to be present, as a result of which the total breeding frame will be changed, so the total production of the protein will be changed. The protein may not be properly functional, the protein may be truncated, the protein may not be functional at all, or may be functional for a limited capability. So as a result of which, what we can do, we can change the transcriptome inside the cell. We can change how exactly the proteins are produced inside the cell. So this sort of transcriptome analysis can be done transcriptome analysis and modification. And modification can be done utilizing CRISPR-Cas9 DNA silencing mechanism. Now, other things are also associated here. Earlier, we only talked about the genome silencing and DNA destruction, but now what we know is that this complex, this is not a single RNA or protein. We have the crRNA, we have tracer RNA, we have the Cas9, we have the PAM site present there. You know, with the activated region of the catalytic units or nucleus domain, we can clip the DNA. But scientists believe that this CRISPR-Cas9 has more potential than only cleaving the DNA. And they were right, because what they did is that now they did some experiment. They will totally shut down the nucleus activity of Cas9. Okay, so we are not going to cut the DNA. The nucleus activity is shut down. Okay, so what will happen? Simply, the CRISPR RNA will allow this total CRISPR-Cas9 complex to be associated and attached with the target DNA. But as the Cas9's catalytic domains are deactivated, it will not cleave the DNA. Now what they can do, they can put additional factors from outside. What kind of factors? Transcriptional activators in case of eukaryotic transcription or eukaryotic cell. Transcriptional activators can be provided, the enhancer molecules can be provided, and as we can provide the transcription activator molecules, they are, and with the help of the NTPs that are present there, they can initiate transcription of this specific gene products in that location. So without designing anything else, by providing a target RNA, we can transcribe our target DNA region inside of the cell, and we can amplify the number of RNA from that stretch of the target DNA, as many as we can. So this is the third very important use, that is, we can allow selective transcription. Selective transcription is allowed. Similarly, we can also add some inhibitory components to prevent the transcription of specific region of the DNA. Means, let's say, along with this CRISPR-Cas9 system, if we add proteins like histone deacetylase enzyme, which will allow the DNA to be recoiled on the histone octamer, then the DNA will not be available for the transcription. So in order to stop the transcription, we can also utilize CRISPR-Cas9 structure along with the other accessory protein to inhibit the transcription. Okay, in this case, histone deacetylase can do that. In both these cases, okay, so allow the specific transcription. Fourth one is inhibition of transcription. Okay, and this is also specific inhibition, means in a stretch of the complete genome, we can targetedly block the transcription of genes, right? This is unique. This was not found earlier. So, you know, we have one molecular tool, that is CRISPR and Cas9, and it can do so many wonderful things, right? So that is why I call it as a Pandora's box for modern biological research or modern molecular biology research, because all these things can be done. One more thing that can be done, simply, this is a hybridization process, right? Because with the help of crRNA, it can interact with the target DNA, right? And that is a hybridization. So what we can do is simply we can use CRISPR-Cas9 system, and again we are deactivating the this PAM sites or deactivating the catalytic subunit of or catalytic regions of the Cas9 protein, and as a result of which, what we can do, we simply tag it with GFP or green fluorescent protein or any sort of fluorescent tag with this protein, because it has a protein and as a template of RNA. So we can attach a green fluorescent protein to that protein or any other fluorescent tag to that protein to mark location of specific genes in our genome. So we can paint, literally we can paint our genome. If you look at the human karyotyping can be done, and also if you want to find out the region and location of specific regions in the DNA, we can mark them with the help of different types and different colors of fluorescent tag with the help of this CRISPR-Cas9 mechanism. So the opportunity is limitless, right? One more thing that I must say, as this CRISPR-Cas9 has a capability of degrading the DNA, we can also use it for the gene therapy processes. let's say a particular gene is not working properly. As a result of failure of working properly, there is a disease formed. So what we can do, we can design a complementary target CRISPR RNA, and we can use it with the CRISPR Cas9 system to target that particular gene, and you can destroy that gene. And if you can destroy that gene, then that disease can be reversed, or the different problems associated of the disease can be reversed. So this kind of things we can also do. Okay, we can incorporate our required nucleotides in specific locations. We can introduce mutations in the DNA. So it is involved in mutagenesis, it is involved in gene therapy. So I don't have literally running out of space to write the importance of CRISPR Cas9. So 6 and 7 you can write it, that it is useful for gene editing in case of the gene therapy. It can be also utilized in case of introducing mutations in the DNA, so mutagenesis process as well. Okay, so if you like this video, please hit the like button, share this video with your friends, and subscribe to our channel to get more videos like that. Thank you. Bye.